Research is the cornerstone of innovation in clinical practice, driving improvements in patient outcomes and shaping the future of medicine. Welcome to the Global Rhinology Network Topical Research Symposium. I'm Ashok Rokade from Winchester, UK, and it is my privilege to welcome you as a host. Joining me as co-hosts are Louise Melia from Bath, UK, and Professor Thibault Van Zele from Ghent, Belgium. This symposium showcases fascinating topics across 4 key modules: General Rhinology, COVID session, and Research in Chronic Rhinosinusitis. A special highlight is the PhD session featuring research conducted by enthusiastic young rhinologists who represent the future of our field. This symposium brings together leading global minds in rhinology who will highlight latest developments with a focus on translating research into impactful patient care. The Global Rhinology Network is dedicated to advancing knowledge innovation. Thank you for being part of this exciting journey. Thanks to Medtronic and Karl Storz for their generous support in making this symposium possible. Welcome to GRN-Topical Research Symposium.
Well, so we have Stijn Vanhee, who's a senior postdoctoral scientist at the University of Ghent with a specific interest in B-cell biology as well as immune receptor profiling. And he's going to be talking to us today on understanding mucosal B-cell responses in allergic airway disease.
Yes, hello. I'll try to share my screen. That works. All right. Are you able to see my screen?
Yes.
Stijn,
Okay.
it's still in the PowerPoint. It's not in presentation mode. Okay, great.
It should be. Okay. All right. Good afternoon, everyone. And thank you to the organizers for inviting me to give this talk, indeed, on the work we're doing on understanding mucosal B-cell responses in allergic airway disease. I'm a little bit under the weather, so sorry if my voice disappears from time to time. So as we all know, the airways are actually the first line defense system against airborne pathogens and allergens, of which we inhale a lot each day. So when we look at the upper respiratory tract, meaning the nasal cavity, and the lower respiratory tract, sorry, the lungs, there's a couple of defense systems there that are put in place. So when we look at a healthy lung, there we have a nice epithelial barrier with tight junctions, which really close the barrier so that nothing can come in. And on top of this, you have a layer of mucosa, of mucus production. So mucus is produced, which will capture a lot of these viruses, bacteria, and allergens that come in. But if there would be a breach in this barrier, there's a whole system of innate immune cells present there to sense the incoming infection or the danger signals from the allergens and to fight it off. Now, this is a normal airway. If we then look at allergic rhinitis, chronic rhinosinusitis, or allergic asthma, there's a couple of things that change. So first, the epithelial barrier gets damaged. So there's, in essence, holes in the epithelial defense layer to which the body reacts. So there's an increase in mucus production. And as I said, this innate layer of the immune system is activated, and it will attract adaptive immune cells. It will attract eosinophils, basophils, you name it, the whole classical cascade to defend the airways. But what we are actually studying is how the the B cell response is governed in the mucosa, because there's a lot of things there that we don't know. Generally, what you would see in the textbooks is that when an allergen or a pathogen breaches this epithelial layer, you will have an antigen presenting cell capturing part of this pathogen or the allergen, taking it to a lymph node which drains the organ. So in case of the lungs, the mediastinal lymph node. And here a reactive B cell to this pathogen or allergen will be activated, after which it will differentiate into a plasma cell which produces antibodies, which can then go through the bloodstream and is secreted into the airway space. However, there's more to it, because in the airways you do find plasma cells that produce antibodies locally, which can be protective or exacerbating, could be IgE, could be IgA. And we don't really know how these cells are formed or how they get there. And to understand this, what we've done is to go to a mouse model where we do a chronic exposure to house dust mite extract. So in this model, the mice are sensitized first to a low dose of house dust mite. And then during the course of 5 weeks, we challenge the mice with a higher dose of house dust mite extract. And this is 3 times a week. And we mainly look at the lungs to look at allergic inflammation or to look at these B cell responses, because as you also saw in the previous presentation, the upper airways of the mice, they're not that easy to get a robust immune response. But when we look into the bronchoalveolar space, so in the airways itself, at the production of antigen-specific immunoglobulins, so we're looking here at DERP2, which is one of the major allergens in house dust mite extract. We see that over time, when we're challenging, the levels of IgG1 and IgA increase in the airways. And with this, when we start to immunophenotype the lung tissue, you always see the comparison here on the left. You see the lung tissue, and here you see the draining lymph node. So you see that in the lung tissue, we get a nice population of plasma cells, which we define as being CD19 negative and Syndicon-1 or CD138 positive. You see that this population is clearly present and also increases in the lung over time. So that's this blue line here. And also memory B cells are really enriched in the mucosal tissue. So here you see a comparison of lung memory B cells versus lymph node memory B cells. And you see here that these go up. It's a percentage of cells, so the starting point at sensitization is not very representative. So looking at this data, we asked ourselves the question of whether mucosal B cell responses upon chronic house dust mite exposure are born in a mucosa. Because the interesting thing is that when we expose these mice chronically to house dust mite, we see that close to the large airways, we get these aggregations of cells. So this is a nuclear staining. So this is aggregates of cells. And when we zoom in on these structures, you can see that there's a nice zonation of B cells and an adjacent T cell structure. So in essence, everything is there to have a functional B cell response in the mucosa. And this is not something that is weird for our mouse model, because when you look in patients with allergic airway disease, you can also find these lymphoid aggregates in the inner wall of the lungs. So here you have the epithelium in green, and here you have a staining for T cells. So this is CD5 staining, and this is HLA-DR staining. So you can see that you really get these lymphoid aggregates close to the epithelium. And also in patients with allergic rhinitis and chronic rhinosinusitis with nasal polyps, you can find these sites of active mucosal B cell responses, where you also find plasma cells adjacent to them. Apologies. So these structures that I showed you, we coin them TLO, or in literature, they're coined TLO for tertiary lymphoid organs. And they're called this way because the the B cell responses you normally look at are governed in secondary lymphoid organs, so like the lymph nodes. And the tertiary lymphoid organs are developing de novo, so they're developing after the secondary lymphoid organs, so they're tertiary. And we call these IBALT in the lung, so it's induced bronchoalveolar lymphoid tissue, over bronchus-associated lymphoid tissue, and INALT in the nasal cavity, so induced nasal-associated lymphoid tissue. And we're really trying to understand what the difference is in labor between the TLO and the SLO. And I would really recommend you to also go or watch the PhD session later. Ines Lammens from our lab is also giving a presentation there, which really shows this division of labor. TLOs, how do they develop? In essence, upon a chronic stimulus or viral infection, signals will be given to the structural cells in the lung, which will then start producing chemoattractants, so that B and T cells will flux into the lung, and they will then self-organize into this dense lymphoid structure. And so also in our chronic model, you can see that we have these have an influx of these B cells, and now we have active germinal center B cell responses, so active sites of B cell differentiation. And we define these by the expression of an B cell activation marker GL7 and the upregulation of FAS on these B cells. And when you compare this to the lymph nodes, so the draining lymph node B cell reactions, you can see that they're actually really enriched in the mucosa of the lungs. And when you look at the kinetics of this, you can see that with time, with increasing doses actually of antigen or repeated doses of antigen, you see that this germinal center fraction is really enriched. So the next thing that we wanted to do is to really understand what is going on there in terms of B cell responses. So what we did is make a high resolution map of lung tertiary lymphoid organ B cells. And for the people who are not acquainted with single cell sequencing, I'll just briefly go through it. So what we do is, from our mice that have been exposed to hazesmide for 5 weeks, we make a single cell suspension of the lung tissue. And we gate out or we remove any cells that were in the circulation, so in the bloodstream, by gating negatively on an intravenous CD45 label. And then we sort out the B cells and we load them onto a system that allows us to do single cell RNA sequencing. So in this example, it's a system from 10x Genomics, the Chromium system. Really sorry for that. So in this system, cells are loaded together with barcode beads, as they're called, into a microfluidics chamber. So that they will end up into a lipid droplet. And here all the RNA of one cell is then tagged so that you still know in the end from which cell it's coming. And in your analysis, you can then discern which cell expressed which genes. I'm having a little bit of trouble with the pointer. Yeah, okay. So when we look at the B cell subsets, we can actually find a couple of populations. And we find here a population of memory B cells, which we define by BAC2 expression. We find a population of plasma cells, which is high in PRDM1 expression. And then we find these active sites of B cell differentiation, which are BCL6 and Ki67 positive. And then these cells here, so the active B cell responses, they express AID, which is the gene that is necessary for class switch recombination. So switching away from the naive IgM, IgD expressing B cells to an IgG1, IgG2, IgA expressing cell. And indeed, when we look at these germinal center responses in the lung, we find both naive cells, but also cells that have switched to IgG1, IgG2C, IgA, and IgE. And so the question is then, do these active B cell responses there lead to something? Well, what we see is when we look using imaging, so this is again the lung of a mouse that has been exposed to hazesmide for 5 weeks. When we stain with CD138, so a plasma cell marker, we find a lot of plasma cells surrounding this TLO structure. And when we stain for the different antibody subclasses, so IgA, IgG1, and IgM, we find these really bright immunoglobulin expressing cells lying around the TLO. So these are plasma cells that are surrounding the TLO. And then one important question is, of course, whether we can prove that these germinal center B cell responses in the TLO are giving rise to the plasma cells in the tissue. And for this, we did single cell B cell receptor sequencing. So on top of the single cell sequencing, we also get the information of the B cell receptors, so the clonal information. And we could show here by a clonal analysis plot. So this plot will connect the different cell types in which clonal types are shared. So if clonal type is shared between the germinal center B cells and the plasma cells, it will put a connection in between. And you see here that there's indeed a connection between these two clusters. And when we enumerate this, when we look at the germinal center B cells, so the active sites of B cell differentiation, we found about half of the clones that we find back there are also found in the plasma cell population. So definitively showing that the lung TLO germinal center reaction gives rise to plasma cells in the tissue. So with this, I think we can conclude that mucosal B cell responses upon chronic hazesmide exposure are indeed born in the mucosa. So they're not just circulating through the bloodstream to then migrate into the lung tissue. And I just want to give you a couple of take-home messages. So we see that chronic antigen exposure induces tertiary lymphoid organs in the airways, and that these tertiary lymphoid organs are local hubs for allergen-specific B cell responses in the lung and possibly also in the upper airways. And we find that these TLO B cell responses are really distinct from systemic immune responses. And as I said, Ines Lammens, who works in our lab, has a short talk this afternoon, and I didn't want to steal her thunder, so she's going to talk about this part of the project. And with this, I just wanted to thank the lab I'm doing this research in. So this is the lab of Professor Bart Lambrecht and Hamida Hamad at VIBU Ghent, and also the Upper Airways Research Laboratory at U Ghent. This VIB single cell core facility, who helped a lot with all these analysis, and the same goes for the VIBU Ghent BioIT team. And if there's any questions, I'm happy to take them. And sorry again for the cough.
Thank you very much for your presentation, especially when you clearly aren't feeling very well. So we really appreciate you coming on and speaking. It's very good of you. I think we're going to move to the next talk and then have a question and answer session at the end of this first session, if that's okay. If you can hold on for a little bit longer, if that's all right. Thank you.
No problem.